Everything you have ever been taught about growing food is built on two rules. Rule 1, plants need soil. Rule 2, plants need sunlight. Every garden, every farm, every greenhouse on Earth obeys those rules. But there is an organism sitting on grocery store shelves right now that breaks both of them. It requires no soil. It requires no sunlight. It eats cardboard, coffee grounds, and old cotton t-shirts and converts them into high-protein food in less than 3 weeks. It produces more food by weight than the material you feed it. And in April 2025, NASA sent it to space because they believe it may be the key to feeding astronauts on Mars. You have walked past it 1,000 times without understanding what it is. Now, you will. The organism is Pleurotus ostreatus. You know it as the oyster mushroom. And before you dismiss this as some niche hobby for mushroom enthusiasts and foragers, understand what we are actually talking about. This is not a plant. It belongs to an entirely separate kingdom of life. It has no roots. It has no leaves. It has no chlorophyll, the green pigment that every single plant on Earth uses to capture light energy and convert it into growth. It cannot photosynthesize. It does not need a single photon of sunlight to produce food. Not indirect light, not filtered light, not a grow lamp, zero light. It is a fungus, and it operates under a completely different set of biological rules than anything else in your garden or on your windowsill. Think about what your garden demands from you. Your tomatoes need 5 months of sun, regular watering, fertile soil, staking, pruning, pest control, and prayers against late blight. Your lettuce bolts in summer heat. Your peppers sulk if nighttime temperatures drop below 55 degrees. Every single crop you grow is locked into a calendar, locked into a climate zone, locked into a relationship with the sun. This organism is locked into none of those things, while your raised beds need compost, fertilizer, irrigation, and 6 to 8 hours of direct sun. This organism needs a plastic bag, some damp straw, and a closet. It grows year-round. It grows in any climate zone. It grows in a New York City apartment and an Alaska cabin with equal indifference. The biological efficiency of oyster mushrooms ranges from 75 to 130%. That number deserves repetition. 130% efficiency means you get back more food by weight than the substrate you put in. No plant on Earth can do that. No animal on Earth can do that. Only fungi can do that. And the reason they can do that is because they are not what you think they are. They are not passive decomposers quietly breaking down dead material. The organism you buy at the grocery store for $4 a pound is a predator. It hunts. It kills with chemical weapons. And the story of how we discovered that begins with a forgotten petri dish in a Canadian laboratory. The archive opens in the late 1970s on the 3rd floor of the science complex at the University of Guelph in Ontario, Canada. George Barron had spent years doing something most scientists considered eccentric, even pointless. He was hunting microscopic predators. Not the kind you would expect, not wolves or hawks. He was hunting fungi that kill. Barron, a Scottish-born mycologist, collected soil samples from fields and forests across southern Ontario. In his laboratory, he grew these organisms on petri dishes, watching through microscopes as tiny thread-like networks trapped and consumed nematodes, microscopic worms no wider than a human hair. These were not passive organisms sitting in the dirt. They were hunters, and Barron wanted to understand how they worked. One dish was forgotten, pushed to the back of a shelf. 6 months passed, maybe longer. The exact timeline is lost to laboratory memory. But when a technician finally found it, something unexpected had happened. The fungus had sprouted a mushroom, not some alien growth. A large, edible, immediately recognizable mushroom, the kind you see in grocery stores every single week. Barron and his graduate student R. Greg Thorn identified it immediately. Pleurotus ostreatus, the common oyster mushroom. To Barron's astonishment, the predator they had been studying for years was already sitting on supermarket shelves around the world. Nobody had known. They published their findings on April 6, 1984, in Science, Volume 224, pages 76 through 78. The paper documented that 10 species of guild fungi actively attack and consume nematodes, but the killing mechanism was what stunned the scientific community. Thorn later recalled watching through a microscope as nematodes encountered the fungus. He described seeing the worm make one last pathetic attempt to wriggle away before going completely still. The weapon was something straight out of science fiction, tiny lollipop-shaped structures called toxocysts, each one smaller than a grain of pollen. When a nematode brushes against one of these structures, it bursts open. The toxin inside paralyzes the worm within 30 seconds. Within 24 hours, thread-like hyphae have penetrated the body and digested it completely. There is nothing left. Barron called them lethal lollipops. This was not a marginal curiosity. This was a foundational revelation. One of the most widely cultivated mushrooms on Earth, sold on every continent, sautéed with garlic and butter in kitchens around the world, is a carnivore. The organism people toss casually into pasta is a hunter that kills with chemical weapons, and it raised an immediate question. If this organism evolved to kill, what else can it do? The answer to that question had already been discovered, not in a laboratory, but in a forest, during the worst food crisis of the 20th century. In 1917, Germany was starving. The British naval blockade had severed the nation's food imports, roughly one-third of its pre-war supply. Conscription had stripped farms of nearly all their labor. Then, the potato harvest failed. Late blight swept through the fields, destroying what should have been the nation's staple crop. The winter of 1916 to 1917 became known as the turnip winter, because turnips were all that remained. Daily rations in some cities plummeted to 1,000 calories. Children collapsed in classrooms, hospitals overflowed with cases of starvation edema, bodies swelling with fluid as organs began to fail. By the time the war ended, an estimated 762,000 German civilians had died of malnutrition. Reports documented 80,000 children starving to death in 1916 alone. In the midst of this catastrophe, a mycologist at the Royal Prussian Forest Academy turned his attention to an unconventional solution. Richard Falck had been appointed the first professor of mycology at the Academy in Hannover-München in 1910. He was a scientist, not a politician, but he understood what others did not. This organism could convert waste materials into edible protein on timescales of weeks, not seasons. And it needed nothing that the war had taken away. No farmland, no fertilizer, no sunlight, no months of growing time. In 1917, Falck published a paper with a title that revealed its urgency: On the Forest Cultivation of the Oyster Mushroom on Hardwood Tree Stumps. This was not abstract research. This was survival biotechnology, written during a famine that was killing thousands of people per week. Falck inoculated hardwood stumps and sterilized straw with oyster mushroom mycelium. He demonstrated for the first time in Western scientific literature that this organism could be cultivated deliberately, not foraged from the wild, not stumbled upon by chance, but grown reliably and repeatedly from materials that would otherwise rot. The implications were radical. Every city in Germany was surrounded by forests full of dead wood. Every farm had straw. Every household had waste material. And in 3 weeks, that waste could become protein. During a starvation winter when crops had failed, fields lay empty, and supply lines were severed, Falck proved there was a third option. You did not need fields. You did not need sun. You did not need rain or seasons or anything the war had destroyed. You needed waste material and patience measured in days. Falck married fellow mycologist Olga Shankelovsky in 1910. They worked together for years. But the story does not end with scientific triumph. After the war, radicalized anti-Semitic students persecuted him. In 1933, when the position he had held for over 2 decades came up for permanent appointment, he was passed over. The reason was explicit: he was Jewish. Falck fled Nazi Germany for the United States. His brother Edward, who had operated a mushroom cultivation business, was murdered in Auschwitz. Richard Falck died on January 1, 1955 in America, far from the forests where he first grew oyster mushrooms to feed a starving nation. Falck proved the organism could be cultivated, but he could not have known what researchers would discover 67 years later: the molecular mechanism that made it all possible. What they found was stranger than anyone expected. For nearly 40 years after Barron's 1984 discovery, the identity of the toxin remained a mystery. Researchers knew oyster mushrooms killed nematodes. They could see the toxicists under microscopes. They could watch the paralysis happen in real time, but they could not identify the chemical weapon the mushroom was deploying. In 2023, a team led by Hsueh, now a director at the Max Planck Institute for Biology in Tübingen, Germany, published findings in Science Advances that answered the question. Hsueh's team had undertaken a massive genetic fishing expedition. They screened approximately 12,000 randomly mutagenized clones of Pleurotus ostreatus, searching for mutants that had lost their killing ability. They found 13. Every single one of them lacked toxicists. The team then identified the active compound. It turned out to be something deceptively simple: 3-octanoate, a volatile ketone, an 8-carbon molecule. When the toxicist bursts, this ketone disrupts cell membrane integrity throughout the nematode's body. Calcium floods into cells that should not contain calcium. The worm's nervous system fires uncontrollably. Muscles lock. It dies from the inside out. Hsueh described it bluntly: nerve gas in a lollipop. But the discovery went deeper. A remarkable finding by researchers in Mexico connected the carnivore to the organism's other superpower: its ability to digest wood. They found that silencing a specific gene called lacadis-2, one of the enzymes oyster mushrooms use to break down lignin in dead trees, eliminated both toxin formation and the mushroom's ability to produce fruiting bodies. The same enzyme system that digests dead wood is required for killing prey. Both processes depend on the same molecular machinery. This is not a Venus flytrap waiting passively for an insect to land. This is not a pitcher plant sitting open. This is active chemical warfare, deployed by an organism you can buy at any grocery store in the country for $4 a pound. And it does this because of where it lives. Dead wood is rich in carbon, but desperately poor in nitrogen. The nematodes crawling through rotting logs are tiny packets of nitrogen, protein wrapped in a worm-shaped container. Carnivore evolved to supplement nitrogen intake. It is directly analogous to carnivorous plants growing in nutrient-poor bogs. Except this strategy uses volatile nerve agents delivered through exploding lollipops instead of sticky leaves or snap traps. More than 700 fungal species are now known to trap nematodes, but the Pleurotus approach is unique among all of them. Now imagine you are a nematode, the width of a human hair, crawling through a chunk of rotting wood, searching for bacteria to eat. The wood is dark. It is damp. It smells of decay, which to you means food is nearby. You are not worried. You are the most abundant multicellular animal on Earth. There are roughly 57 billion nematodes for every living human. You have been successful for 500 million years. You brush against what looks like a tiny ball on a stick, no bigger than a speck of dust. That ball explodes. The air around you fills with a volatile chemical, an 8-carbon ketone that begins dissolving your cell membranes on contact. Calcium ions flood your muscle cells. Your body contracts involuntarily. You try to move, you cannot. Within 30 seconds, you are completely paralyzed, but still alive. Over the next 24 hours, thread-like filaments called hyphae penetrate your cuticle, branch through your body cavity, and digest you from the inside out. Every molecule of nitrogen, every amino acid, every trace of protein in your body is absorbed into the fungal network. There is nothing left. You are food. And the organism that ate you is the same one that humans buy in a plastic container for $4 at the grocery store and toss into a stir-fry. This organism does not just eat wood. Research confirms it can convert coffee grounds, cardboard, straw, old cotton clothing, sawdust, corn cobs, newspaper, even diesel-contaminated soil into edible protein. The protein content is 20 to 30% on a dry weight basis, and it contains all 9 essential amino acids. It needs 1.8 gallons of water to produce a pound of mushrooms. A pound of beef requires roughly 1,800 gallons. 1 acre of mushroom production can yield up to 1 million pounds of food per year. 1 acre of cattle pasture yields roughly 300 pounds. The carbon footprint is 0.7 pounds of carbon dioxide, equivalent per pound of mushrooms. A pound of beef generates roughly 14 times that. The numbers are not subtle, they are staggering. And in 2025, NASA took notice. Researchers on the International Space Station successfully cultivated a sporeless strain of oyster mushroom on substrates made from cotton t-shirts and inedible plant biomass. Worn-out crew clothing that would normally be compacted and burned up in Earth's atmosphere was instead turned into food. A 70% cotton and 30% soy hull mixture outperformed traditional straw-based substrates. The harvested mushrooms were safe to eat, with bacterial counts well below commercially grown specimens. Then in April 2025, the SpaceX Fram-2 mission carried mushroom substrate into polar orbit in an experiment called Mission Mushroom. An Australian astronaut monitored fruiting bodies developing in microgravity, making it the first time mushrooms were deliberately cultivated in space. NASA now ranks food and nutrition as the 11th most critical challenge for deep space exploration. Ralph Fritche, former senior project manager of NASA's Space Crop Production Program, admitted something remarkable. Fungi were the clear winner for sustainable space food systems. A team from the University of California, Riverside, called No-Lux Slux, pushed even further. Using a process called artificial photosynthesis, they converted exhaled carbon dioxide and water into oxygen and acetate through electrolysis. The acetate was then pumped into a growing chamber to feed oyster mushrooms growing in complete darkness. No soil, no sunlight, no traditional substrate. Just human breath and water, turned into food. Their findings were published in Nature Foods. The process is more efficient at converting energy into biomass than natural photosynthesis, but the very process that I already see that every plant on Earth depends on. Think about that for a moment. Scientists have found a way to grow food more efficiently than photosynthesis itself, and the organism that makes it possible is the same one sitting in the produce aisle of your local supermarket. If NASA trusts this organism to feed astronauts on missions to Mars, missions where every gram of cargo costs thousands of dollars, and sunlight is roughly half as strong as on Earth, the question becomes unavoidable. Why are you not growing it in your kitchen right now? The answer is not a conspiracy. There are no secret memos. The suppression is older and stranger than that. In 1887, British mycologist W.D. Hay wrote something prophetic. He observed that the fear and hatred of fungi is not a human universal. It is merely British, American, Canadian, and Australia. In the 1950s, mycologist R. Gordon Wasson and his wife Valentina Pavlovna Wasson formalized this observation into a framework. They divided cultures into 2 categories: mycophobic cultures that fear and avoid fungi, primarily Anglo-Saxon societies, and mycophilic cultures that love them, such as Russian, Eastern European, Italian, Chinese, and Japanese. The term mycophobia entered the Oxford English Dictionary in 1957, not as slang, but as a recognized cultural condition. The cause is traceable. Researchers have linked it specifically to industrialization. When the Industrial Revolution pulled massive populations from rural villages into factory towns, oral foraging traditions shattered. For thousands of years, mushroom knowledge had been passed from grandmother to grandchild, from neighbor to neighbor, through direct hands-on experience in forests and fields. But grandmothers who knew which mushrooms were safe did not follow their grandchildren to the mills of Manchester and the factories of Philadelphia. The knowledge was not written down. It lived in people's hands and eyes and memories. All it takes is one break in the chain, one generation that moves to a city and never walks through a forest with an elder. That broken link became codified into institutions, textbooks, regulations, and a deep cultural assumption that all fungi are dangerous until proven otherwise. The irony is astonishing. Today, only 2% of known mushroom species are actually toxic. Yet Anglo-Saxon culture treats the entire kingdom, all 15,000 known species, as suspicious. Meanwhile, in Russia, families spend weekends foraging mushrooms the way Americans spend weekends at shopping malls. In Italy, fresh porcini mushrooms are sold at open-air markets with the same casual trust that Americans apply to apples. In China and Japan, mushroom cultivation is a multi-billion-dollar industry with centuries of accumulated expertise. The fear is not human, it is cultural. And it was installed in your ancestors less than 200 years ago. The economics reinforce the amnesia. The global mushroom market is valued at over $73 billion in 2025, growing at roughly 10% per year. The oyster mushroom segment alone is growing at 11.5% annually, but the global meat industry generates over $1.4 trillion. Mushrooms are dwarfed by a factor of 20. You cannot patent an organism that grows on waste. You cannot create input dependency around it. There are no seeds to sell every season, no proprietary fertilizers required, no herbicides to spray. Mushroom production is labor-intensive, which makes it resistant to the kind of industrial scaling that creates corporate empires. The American Mushroom Institute is a footnote beside the National Cattlemen's Beef Association. There is no suppression required. The sheer economic mass of the incumbent protein industry creates an asymmetry that no mushroom lobby can match. Fresh mushrooms are highly perishable. You cannot ship them globally, like frozen beef. You cannot warehouse them for months, like grain. The shelf life is measured in days, which means production must be local, which means it resists the centralization that creates billion-dollar corporations. They benefit from a population that fears fungi, that sees mushrooms as dangerous and marginal, that builds its entire concept of protein around fields and feedlots and supply chains, requiring 18 months and thousands of liters of water, not 3 weeks and a cardboard box. They have built an entire civilization around the idea that growing food requires land, sunlight, water, and time. The organism that proves otherwise gets no advertising budget, no lobbying arm, and no shelf space in the cultural imagination. But forgetting is not permanent. What was put away for a while can be found again. In 2009, 2 college seniors at the University of California, Berkeley, were sitting in a business class when their professor mentioned something in passing. Mushrooms, he said, could grow on coffee grounds. Nikhil Arora and Alejandro Velez were studying business and economics. They had corporate job offers waiting. But that single sentence would not leave them alone. They started experimenting in their fraternity house kitchen, collecting used coffee grounds from campus cafes, waste that would have gone straight to landfill. They filled plastic containers with the grounds, mixed in oyster mushroom spawn they had ordered online, and waited. They did not know what they were doing. They had no mycology training. They had never grown anything. But within weeks, mushrooms were erupting from garbage in a fraternity house in Berkeley, California. That moment changed both of their lives. They built the first prototypes of what would become their signature product, a cardboard box kit that produces up to 1.5 pounds of oyster mushrooms from 2 harvests in roughly 10 days. Everything you need comes in one package. Open the box, spray it with water twice a day, harvest in a week. The setup takes less time than making a pot of coffee. When the corporate job offers came, Goldman Sachs and the consulting firms and everything their degrees had prepared them for, they turned them all down. They were encouraged early on by Alice Waters, the woman who arguably started the farm-to-table movement in America through her restaurant Chez Pinese. Back to the Roots has since grown into America's #1 organic gardening company. Their products are sold in over 10,000 stores nationwide, including Home Depot, Target, Walmart, and Whole Foods. They have diverted over 1 million pounds of coffee waste from landfill and donated growing kits and educational materials to more than 10,000 classrooms. But the revival extends far beyond Berkeley. In Rotterdam, a company called Rotterdam collects coffee waste by cargo bike and produces 1,200 to 2,400 kilograms of oyster mushrooms monthly from a converted tropical swimming pool. The building was abandoned. The coffee was garbage. the mushrooms turned both into a business. Mark Schlegers, the co-founder, described their philosophy simply: nature makes no waste. If you look at nature and learn how it works, you can make a profit. In Devon, England, an operation called Grow Cycle ran the United Kingdom's first urban mushroom farm from the third floor of an empty office building, collecting coffee grounds weekly by cargo bike and turning them into food in a space that had sat vacant for years. In Brooklyn, New York, a company called Smallhold grew mushrooms in modified shipping containers tucked beneath the Williamsburg Bridge, producing tens of thousands of pounds per week, in a space smaller than a typical studio apartment. Mycologist Paul Stamets framed the larger vision when he said every community should have a gourmet mushroom farm to build carbon in the soil, provide local healthy food, and recycle proximate sources of debris and waste. A 2025 study from Cornell University's Department of Plant Pathology found that oyster mushrooms have a 94% success rate among first-time growers, the highest of any variety tested. This is not difficult. This is not advanced mycology. This is the single most forgiving food production system available to a human being. So here is exactly how you start. You need 4 things. First, pasteurized straw or cardboard. Cut the straw into 3 to 4 inch pieces or tear the cardboard into small strips. Soak it in water heated to 160 to 180 degrees Fahrenheit for at least 2 hours. This kills competing organisms without sterilizing the substrate completely. Drain it until it is damp but not dripping. Second, grain spawn. Available from suppliers like Fungi Perfecti, North Spore, or Field and Forest Products, a 5-pound bag costs roughly $20 to $25 and will inoculate multiple batches. Third, a container. A perforated plastic bag works. A 5-gallon bucket with 1/4-inch holes drilled every 6 inches works even better. Fourth, an environment between 60 and 75 degrees Fahrenheit with some humidity and fresh air exchange. That is it. No soil, no grow lights, no fertilizer, no garden bed. A closet works. A garage works. Under your kitchen sink works. A spare bathroom works. Layer the damp substrate and spawn in your container like a lasagna. 1 inch of substrate, a sprinkle of spawn, another inch of substrate, another sprinkle. Fill it up. Close the bag or put a loose lid on the bucket. Place it somewhere dark with a stable temperature. The timeline is precise. 12 to 21 days for the spawn to colonize the substrate. You will see white thread like mycelium spreading through the straw or cardboard, like a slow-motion web. Keep it in darkness during this phase. Mushrooms do not need light to grow their root network. Then move it somewhere with indirect light and fresh air. Within 3 to 5 days, tiny pin-like structures will appear where oxygen reaches the substrate through the holes. These are baby mushrooms. Then 4 to 7 days from pins to full harvest size caps. Your first harvest can happen in as few as 19 days from setup. And here's the part that separates this from everything else in your garden. After you harvest, the mycelium is still alive inside the substrate. It is not done. It will fruit again. 3 to 4 successive flushes are typical, spaced 7 to 14 days apart. Between flushes, soak the substrate in cold water for a few hours to rehydrate it, then put it back. One 5-minute setup gives you a month or more of recurring harvests from the same container, the same bag, the same bucket, producing food over and over again with nothing added except water. On optimized substrates like coffee grounds or cotton waste, biological efficiency reaches 130%. More food out than substrate in. No plant in your garden will ever do that for you. No seed you will ever buy comes with that promise. When the substrate is finally spent after its last flush, it becomes excellent compost for your garden beds. It has already been broken down by some of the most powerful decomposition enzymes on Earth. The mushrooms feed you, and then their leftovers feed your soil. Nothing is wasted. If you prefer to skip the setup entirely, pre-made grow kits from Back to the Roots are available at Home Depot, Target, Walmart, and Whole Foods for around $15 to $20. Open the box, spray it with water twice a day, harvest in 10 days. Every kit comes with a 100% guaranteed to grow promise. If you want to forage wild specimens, oyster mushrooms grow on dead or dying hardwood trees across every temperate continent. They fruit primarily from October through April in North America. Look for fan-shaped clusters on oak, maple, beech, or poplar logs, but you must know how to distinguish them safely from their most dangerous look-alike, angel wings, Pleurotus porogens, which was linked to 17 deaths in 2004. 3 features separate them reliably. First, oyster mushrooms grow exclusively on hardwood. Angel wings grow on conifers. If it is growing on a pine or spruce, do not touch it. Second, oyster mushrooms have thick, fleshy caps. Angel wings are thin and translucent, almost paper-like. Third, take a spore print. Oyster mushrooms produce a lilac gray print. Angel wings produce pure white. Always confirm identification before eating wild specimens. When in doubt, leave it alone. But understand what you are really doing. When you grow this organism, you are not gardening in any traditional sense. You are activating a biological technology that was refined across 100 million years of evolutionary pressure in rotting forests and handed to you as a system that converts waste into food without soil, without sunlight, without fertilizer, without pesticides, and with a fraction of the water that every other food source on Earth demands. The cardboard box in your recycling bin is, to this organism, a steak dinner. The coffee grounds in your trash can are a feast. That bag of straw in your garage is a protein factory waiting to be switched on. Every assumption your garden runs on, soil, sunlight, water, space, time, this organism bypasses all of them. It is the only major food source on Earth that a person can produce in a closet, in total darkness, from materials pulled out of a recycling bin, with a harvest cycle measured in days instead of months. They did not steal this knowledge through conspiracy. They lost it through cultural amnesia and economic indifference. 108 years ago, Richard Falck proved it could feed a starving nation during the worst winter in German history. He was driven out of his country for being Jewish and died in exile, but the knowledge survived. 41 years ago, George Barron proved this humble grocery store mushroom was a predator armed with chemical weapons that would make a military chemist pause. 2 years ago, Hsueh identified the nerve agent inside its exploding kill structures, a volatile ketone so elegantly simple that it uses only 8 carbon atoms to dismantle every cell in a nematode's body. Last year, NASA grew it on cotton t-shirts aboard the International Space Station and declared fungi the most promising food source for deep space missions. This year, it became the first food organism deliberately cultivated in orbit around Earth, circling the planet at 17,000 miles per hour, while quietly converting waste into protein the same way it has for 100 million years. And yet, walk into any garden center in America and ask for help growing mushrooms. Watch what happens. You will get blank stares. You will be pointed toward the outdoor plant section. The entire infrastructure of home food production, the seed racks, the soil bags, the fertilizer shelves, the grow lights, all of it is built around plants, around soil and sunlight. The organism that breaks both rules has no shelf space, no aisle, no section, not because it does not work, because we forgot. The organism does not need you. It has been converting waste into life for 100 million years without human help. But you might need it. And now, you know exactly how to use it. If you found this fascinating, subscribe for more stories like this. If you have grown mushrooms at home before, or if this is the video that finally convinced you to try, tell me about it in the comments. The setup takes 5 minutes. The first harvest takes less than 3 weeks. Once you see mushrooms erupting from a bag of straw in your closet with no soil, no sunlight, and no garden required, everything you thought you knew about growing food will change forever.